Hi guys, welcome to another instalment of Biology with Miss Cherry from home. As last time, you'll have this video. You can pause it at any time, complete the tasks, go back, rewind me. But when I say pause, please don't skip the video on to forget the answers. Specifically talking to some of you, not saying that you're all cheaters, but I know that some of you are. So this is the end of Unit 2. We're going to be looking at starting to understand how the body fights infections. So for now, pause me. You've got a couple of minutes just to recap what you know, do these remind me questions, and come back in a couple of minutes or so. So, thinking about the three major types of infectious pathogen, hopefully you will have got bacteria. Hopefully, I'm going to change pen. Apologies, I'll go straight to black. You would have got viruses, and hopefully you will have also got fungi or fungal diseases. Now you might know that our body fights against infection using the immune system. You might have mentioned something about phagocytes or phagocytes, depending on how you want to say it. You might have said about white blood cells. You might have had some more detail, and if you do have more detail and you want to add it to the collaboration space on the OneNote class notebooks, especially for my class, you are more than welcome to do that, and we can discuss things there. Okay, now what you might not know, and what we're going to look at over the rest of the lesson, is how our body distinguishes between self and non-self cells, and that's all to do with the proteins on the cell surface membrane. Okay, so cell surface, apologies for that horrible R, membrane, which I'm going to abbreviate to CSM because I am lazy, and these proteins are called antigens. Okay, we discern between self and non-self antigens by looking at the shape of those proteins. So again, you might have had different points. You might decide to completely ignore the instructions from the beginning of this video and have just skipped to this point, in which case shame on you. But if not, we're going to get started with the lesson. So this is lesson 2.16, defense and phagocytosis or phagocytosis. We're going to be looking at the main defense mechanisms of the body. We're going to look at self and non-self. We're going to have a look at phagocytosis or phagocytosis, and then look at the role of lysosomes in phagocytosis. Lysosomes, you'll remember, are the membrane-bound vesicles that we find inside our cell. If you're not sure about that, maybe you should take a look over the cell organelle stuff from the beginning of Module 2. Now, thinking back to GCSE, I know you didn't do your exams, but we've still got to be aware of the fact that communicable diseases are those that can be passed on. We say they're infectious. They can be communicated between things. Oh, if I could spell, that'd be great. Communicated between organisms. An example would be coronavirus, which is exactly the reason why you're at home. Okay, that's an example of a communicable disease. It's a viral particle. Maybe an organism is a little bit too far, but it infects us and we are organisms. Okay? Generally, we say that these communicable diseases are caused by pathogens, which are your bacteria, fungi, and viruses, but also for A-level, you need to know about protists. These are pathogens. And protists are a sort of different type of cell. If you want to look at a little bit more in detail, do your own research, or it might be in the textbook. I'm not too sure because I haven't got one in front of me right now. Now, the pathogen itself, you need to be able to define as a disease-causing organism. And this should hopefully be on your keywords list. If you're not too sure, take a look, if you're in my class, at the content library, and it should be under 0 admin, and I've put a link to the keywords there. If you're Mr. Mahita's class, just ask him to send you the file, and I'm sure he'll be able to. Now, infectious diseases, pathogens, communicable diseases, all of that stuff are one of the leading causes of death worldwide. If you think about things like malaria, COVID, flu, other infectious diseases, things like HIV that we're going to look at towards the end of the lesson, they cause a lot of deaths. Even if we're thinking about COVID now, there's been in excess of, 50,000 deaths I think we're at right now. That's quite a lot. Yeah, so it's a vast proportion of the society. But fortunately, we're able to protect against pathogens, and we've got two different ways that we do it. Okay, the way that we protect against pathogens is called our immunity or our immune response, and it can be separated into two separate branches. So it can either be non-specific, a sort of general fight everything as soon as it arrives kind of situation, and this is including your phagocytosis and physical defences. These are things that are found on your body and help to protect you. Now this is a day-to-day thing. Okay, your non-specific defences are always there. And if they aren't, maybe you've got a genetic mutation that means that you maybe don't have one of these, then you're in a little bit of a sticky situation. Conversely, we've got the specific defence mechanism, and this takes a little bit longer. Okay, it's a little bit longer to activate, but it's longer lasting as well. And because it's called the specific defence mechanisms, it's targeted to the pathogen. Okay, so in this, we make specific cell types called B cells and T cells that are specifically shaped to attack whatever pathogen is infecting us. And this is really important because this gives us things like our immunity to things that are around us all the time, things like colds, things like the flu. Okay, so for this lesson, we're going to take a bit more of a look at the non-specific defences, and we're going to start by looking at the immune system. Okay, so the immune system we've said over here is made of the non-specific on the left branch and specific on the right. And we say that this immune system, the non-specific immunity, is our first line of defence. Okay, first line is generally your physical responses, and I don't really like this second line, but we'll put it there because it's there. Second line of defence is normally your cellular responses. Okay, this is normally after things have gotten into your body. So things like inflammation, if you get a splinter and it gets really hot and sore and pussy, that's because of the inflammatory response trying to push it out. The specific immune response, though, on the right-hand side, is what we spoke about. That's that slightly longer to act, slightly bit more faffy, cell-based response. Okay, specific and specialised to the pathogens. Now, considering those physical barriers, you can see from this diagram there are lots and lots of different barriers that the body has that you might not have even thought about. And we're going to spend a little bit of time over the next few slides looking at those in a little bit more detail. So if you're confused at any point, if I talk too quickly, just pause, make some notes, and then carry on playing. Now, the major one of our physical defences that is most often broken is the skin. Okay, the skin is the largest organ on the body. We know it's an organ because it's a collection of tissues working for a single function, and it produces sebum. Okay, now, sebum is normally the thing that on your face and on your body blocks pores, and in any other situation like your face and things like that, with the blocked pores, they would produce things like spots. Now, it's not to say that it's just there to produce spots, that'd be really annoying otherwise, but actually what it does is provides an oily environment that stops pathogens growing. Okay, it inhibits their growth by reducing the conditions, maybe the aerobic conditions that are there to help those pathogens respire. So it's not there just to annoy you, it's there to actually do something useful. Also on your skin, we've got flora. Okay, now flora is the thing that we use to describe the colonies of bacteria on the skin. Okay, now that's not to say that all of you are disgusting and dirty. They're natural, they're there, and the idea is that they take up space and aim to outcompete the pathogens for surface space. Okay, so if the pathogens can't sit on your skin's surface, they can't access nutrients from the cells, they can't access maybe things like water that's coming out of your pores, things like that. And if the pathogens don't have space to reproduce and survive, they're not going to be able to survive on the skin. Inside our body, we've got mucous membranes. Okay, mucus is the thing that gives you a little bit of phlegm, but it's also there as a kind of sticking agent. Now, generally, we say that the mucous membranes are found on the gas exchange surfaces. Things like your trachea, maybe your bronchioles, or your bronchi. And what it does is it traps the pathogens, okay? So it's gunky, it's secreted from goblet cells. And that trapping of the pathogens means that they can't get into the gas exchange system and they can't cause things like respiratory infections. Now, the good thing about these mucous membranes as well is in trapping the pathogens, they enable lysozymal digestion. Okay, so remember when we're thinking about our suffixes, YMES is an enzyme. Okay, so lysozymes are a group of digestive hydrolytic enzymes that we find in lysosomes. Okay, and we'll note they're hydrolytic. Now, what that means is they work by helping those pathogens undergo hydrolysis reactions, breaking stuff apart using water, and they basically bust up the pathogen. So they stop it functioning, they maybe put some pores in the cell membrane, things like that. The water potential differences cause water to move in or out of the cells depending on the situation, and they will burst or shrivel, blah, 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 blah. You just need to know that lysozymes are there. Now, those lysozymes are actually found in multiple places, not just the mucous membranes, not just the mucous membrane cells, the goblet cells, for example. They're also produced by things like tears, okay? Tears and urine and stomach acid, okay? Things that are there as easy passages in and out of the body. Okay, remember when you're eating things that are bad, maybe you're eating a little bit of cheese that's a little bit blue because you're a poor student, which you'll be in a couple of years. That stomach acid is going to help destroy the pathogens and absorb them with the lysozymes, or digest them, sorry, with the lysozymes, and then they just get absorbed and recycled. Now, we said that we're going to look over the self and non-self cells. And the self and non-self cells are distinguished by lymphocytes. Okay, lymphocytes is just the sciencey word for white blood cells. Sometimes they are also called leukocytes. So if you know anyone who's got leukemia, for example, that's a cancer of the white blood cells, leuk. Okay? Now, these lymphocytes or leukocytes have to be able to distinguish between your cells and cells that are dangerous or bad or non-self. Okay, so if I start down the bottom, imagine this is my cell. Okay, so a nice smiley happy one. Ha 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 ha. And on the outside of my cells, I've got antigens, proteins that are a specific 3D shape. Okay, so they're 3D shaped proteins. Now, because they are 3D shaped, they have got a tertiary structure. Okay, they require a lot of folding. And my body, my lymphocytes, are able to bind using the receptors for self onto them. So this is my white blood cell, just checking whether it's me or not. But sometimes a bad cell will come in, a pathogen maybe, with different shaped antigens. Okay, so maybe a pathogen has got slightly more rectangular shaped antigens. And in that case, the white blood cell will bind to it with a different set of receptors, like that. Okay, that's a horrible picture. I'm going to rub that out and start again. So the white blood cells will sort of tootle on. This is a question mark, and bind to those antigens with a different type of receptor. So if our white blood cells have receptors for self that bind to self cells, they're like, okay, cool, that's fine, carry on. But if the receptors for non-self bind to a pathogen, then your body's like, hell, let's start to respond. And sometimes that response is an immune cascade. So all of this slide says is what I did in a horrible diagram on the previous slide. So what you can do if you're not too sure is you can just pause me now, take a look at this information and draw your own diagrams. Okay, I'm not going to spend too long on this slide because it's exactly what I've just said. Now, recognising between self and non-self is not just as simple as recognising pathogen cells or pathogens in the body. We can recognise those pathogens, but we can also use this self-non-self recognition to recognise different materials in the body, which isn't always helpful. So an example might be if you've got a transplant. Maybe you've had a kidney transplant because you've had kidney failure, and that's going to come from someone else. And if the body recognises that you've got a cell or a set of cells in an organ that isn't your body, again, they're going to be like, oh, hell no. So this is why people who do transplants and stuff have to take immunosuppressants. So they want to suppress their immune system, reduce the immune response, so we don't get that kind of self-non-self recognition. Now, helpfully, the body is able to recognise non-self in terms of things like toxins, so bacterial toxins that are produced to give us those symptoms. And also we can recognise abnormal body cells, so things that have maybe mutated during cell division, maybe things that are going to become cancerous. Okay, and the body will try to recognise them as non-self and destroy them. Now I want you to pause and have a think about these questions. Okay, as we've seen, the immune system is specialised to help us fight off any non-self cells, even if they are helpful to us. Could you please, with regards to transplantation, so the addition of organs from another source into your body, how and why, and that's the important bit, the why is also important, does the immune system respond to foreign tissues in the body? And how do doctors try to minimise the effect of the immune response post-transplantation? So pause me now, spend a couple of minutes, and then play again, and we will come back to it. Okay, so hopefully you've spent a little bit of time and haven't just skipped and, not followed the instructions, which is not unlike some of you. But hopefully you will have been able to recognise that the body's white blood cells, you could have said lymphocytes or leukocytes, recognises non-self cells when non-self receptors bind to the transplanted cell. Okay, this triggers the immune response. Remember, it's protective. I'm sorry for my awful handwriting. I hate this pen. Okay, so how the body's white blood cells recognises the non-self cells when the non-self receptors bind to the transplanted cell, and this triggers the immune response. Now, why it does this, as we've said, is it is protective. Okay, we don't want things inside of our body that are dangerous. And the way that we try to minimise the effects is, firstly, we try to match the antigens as closely as possible. So this is why you might have known that if you have a transplant, they try to see if there's anyone close in your family that has the same sort of blood type and antigens as you, so that it's less chance for the body to reject them. So we say family or siblings. And secondly, as I said on the previous slide, we're going to use immunosuppressants. I don't know if that's how you spell it. I'm going to go with it anyway. And that's special medication that reduces the immune system to stop responses happening. Okay, they're drugs that you have to take for life, but I suppose it's a thing that you're willing to accept if it means you can live a little bit longer, I guess. Now we're going to have a look now at moving into the specific response. Okay, and for the specific response, we're looking at lymphocytes. Okay, lymphocytes are a type of white blood cell And generally, these come from your mum. Okay? As soon as you're conceived in the womb and you develop into a fetus, your mum's blood is going to be coming and mixing with your blood, and she's going to provide a specific set of cells that she's been exposed to, those lymphocytes, that give you immunity from the very beginning of your life. Okay, and the way these lymphocytes work is that lymphocytes also have receptors on them. Okay, all cells have receptors, and as you grow up, develop, experience different pathogens, lymphocytes develop that contain receptors that are complementary or specific to pathogens. Okay, and when those, when you get your lymphocyte, which I'll draw down here, lymphocyte and pathogen, lymphocyte, when they fit together in a weird biological jigsaw and they're complementary to each other, that triggers the cascade of the immune response. Now, lymphocytes work by the antigens binding to the lymphocyte, which is that cell in the diagram, and undergoing something called clonal selection. Okay, clonal selection and clonal expansion. Now, clonal selection means that in this diagram over here, you can see that there's lots and lots of different cell types. Maybe there's like cell type A, cell type Z, cell type B. But this antigen is antigen X. Okay? It only binds to the lymphocytes with the receptors for it. Okay? Now it binds to those receptors on the lymphocyte X here, and clonal selection means that that lymphocyte has been attached to and selected. Now, clonal expansion is what you can see down here, and that's where we get the production of lots and lots and lots and lots and lots and lots of that same lymphocyte. Okay, we're going to expand the number of clones of that lymphocyte that we have. So we're going to increase numbers of specific complementary lymphocyte. Okay, and in doing that, our body is able to either start producing antibodies if it's a bacteria cell, for example, or they can start flagging up the pathogens for destruction through things like phagocytosis, which we're going to have a look at in a second. Didn't mean to do that. But before then, could you please use your books, because I know you've all got them, and hopefully, because you haven't had to lug them anywhere, you should be able to find them. Go to page 103 in your big blue books, the Oxford ones, not the green revision ones. And at the bottom of page 103, there's a set of bullet points. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 bullet points that explain how lymphocytes recognise cells beginning to the body. Could you please pause now and spend 5 minutes or so summarising those bullet points and come back when you are done? Okay, I'm not going to go over it, so you better do it. Better do it. I'll know. When I ask you a question, I'll know. Now, the non-specific defences that we're going to take a look at are looking at phagocytes or phagocytes. I say phagocytes. You don't have to say it that way. I don't really care either way. It doesn't matter as long as you can spell it. Now, phagocytes are a type of white blood cell. They're a type of leukocyte, lymphocyte, and they make up that secondary line of defence. They are still non-specific. Okay, if you imagine the phagocytes are a bit like me with cake. I don't really care about any cakes. I'll eat them all. Yeah, so it's what happens after the pathogen has gotten through those physical barriers through the first line of defence. Okay, secondary line of defence is where we get this engulfing motion happening. So what the phagocytes are there to do are to engulf and remove via digestion any foreign matter. That could be a little bit of debris. Maybe you've, I don't know, licked a pencil and you've got some lead or whatever, graphite in your tum-tum, and it's decided that that's not good because it isn't good. So don't lick pencils if you are. and it digests them. Okay, any foreign matter. Toxins, bacterial cells, cells that are infected by viruses if they've been broken down. Okay, anything, doesn't matter, going to engulf them and digest via phagocytosis. Now, phagocytosis itself, as a definition, is engulfing matter within a vesicle. Remember, they are membrane-bound sacs. Because we're talking about membranes, we're specifically looking at eukaryotic cells. It would be weird if pathogens like bacteria, which are prokaryotic, had things to break down other pathogens, wouldn't it? So we're talking about eukaryotic cells. And the way we engulf is by a process called endocytosis. So we're going to take substances in, a bit like taking them in a hug, and that enclosure of the membrane sticking out, engulfing into a hug, endo taking in, and cytosis is to do with that vesicle itself taking into the cell. All right, so here we've got a diagram of what happens in phagocytosis. Okay, could you please spend some time looking over the diagram and making your own word and picture, if you fancy, step by step of phagocytosis. I'm going to go over the steps and the detail that you need on the next slide. But in the meantime, spend some time, do some doodling. Know you love artwork. Kills a bit of time and means you don't have to listen to me as well for a little bit. And have a go at that. Play when you're ready. So in terms of content, the words in bold are the key words that you need. Okay? So pathogens or damaged abnormal cells, toxins, whatever, are going to produce a type of molecule called a chemoattractant. Chemo is a chemical, and attractants are just going to draw cells to them. Okay, so draw cells towards it. Okay, and that is going to work by effectively all of these pathogens and stuff where in a great big hive is going, look at me, I'm here. Okay, those phagocytes are then going to go to that area of the body and they're going to recognise the foreign proteins, those antigens, on the pathogen cell and attach to them via receptors. Okay, remember, when we're talking about things binding, we have to say that they are complementary. They fit together. Not complementary. We're not talking about them going, oh, you know what, you're so nice. We're talking about complement. They complement each other. They bind together. Now, what we get happening is that phagocyte engulfing. Okay, it wraps around the pathogen, a bit like a hug, and pulls it in. That endocytosis that we spoke about. Now, in doing this, the engulfing, the pulling it in, the pathogen is put into something called a phagosome. Okay, so a phagosome is a lysosome or a vesicle. I'm going to just say lysosome, which we know is a vesicle, in a phagocyte. Okay, so it's being a bit more specific about what that cell or what that vesicle is. Okay, now the phagosome with the pathogen, nice and included in it, then binds with the lysosomes. And those lysosomes we know contain lysozymes. Now together, the phagosome with the lysosome, phagosome plus lysosome, forms a phagolysosome. Now they've just stuck the two words together. Nothing dramatic, no major changes there. But the lysozymes inside of the lysosome, so zyme for enzymes, inside of the some, the vesicle, are going to digest the pathogen. We've said they're hydrolytic. We're breaking it down using hydrolysis reactions, the addition of water, and it breaks things down to bust up that pathogen and make it not 3D anymore, which is fantastic. We don't want it 3D. And once we've got that happening, everything that's inside that phagosome, the digested pathogen, for example, is diffused into the cytoplasm of the phagocyte, and then this is where we start looking at the specific immune response in a bit more detail. But that's next lesson. We're not going to worry about it for now because there's loads of hoo-ha after that. Okay? So we're just going to stop with the digested pathogen in the phagosome being diffused into the cytoplasm of the phagocyte. If you aren't sure about any of this, now's the time to pause this video and talk to either me or Mr. Mahita, depending on which of you is our teacher. Obviously, if you're not in my class, I can't recognise your voice. Don't ask me a question, but you can email me. Okay, so pause now, recap, and then come back when you're ready. All right, guys, so you might want to spend some time now looking over the notes from the previous slide, maybe recapping, going back, repausing, and make a better, more refined cartoon strip or diagram just to show the process of phagocytosis a little bit more clearly. Might help if you're quite visual. Maybe you want to be able to remember that process inside your head through a series of pictures for any assessments that come up. Now, this is a little bit of a pit stop. I want you to spend some time thinking about these questions, thinking about them, writing them down. You can discuss with me or Mr. Mahita if you want during your lesson. That's absolutely fine. So, question 1, lysozymes, as well as lysosomes in phagocytes, are also found in tears. And I want you to suggest a reason as to why we might find those lysozymes, those hydrolytic enzymes, in tears. And then state 2 differences between a specific and non-specific defense mechanism. So, pause and come back. Now, lysozymes are found in tears because the eyes are very close to a really important organ that you might not have heard about called the brain. So we want to digest any any pathogens that are close to our eyes so we don't damage them, so they don't get into our bloodstream, so they don't go to our brain. Okay? Visual or sense organs. Now, hopefully, if we can destroy everything before it gets into our eyeballs, then we should be a little bit protected. And 2 differences between a specific and a non-specific defense mechanism. Non-specific doesn't have a target. Non-specific works for all pathogens or foreign material. Is our first line of defense. Say maybe it's quicker or more readily available. And the converse is true for the specific mechanisms in that they are slightly slower, they're not as readily available, they do have a target, they don't work for all pathogens. Remember, we're looking at that clonal selection and then clonal expansion, and it has to be part of the 2nd or 3rd line of defense. If you're not sure, you've got any questions, ask one of us and we'll get back to you. Now, the final thing that we're going to look at is the bit at the end of that phagocytosis diagram that we saw a few slides back. And this is a specific type of phagocytosis that occurs with macrophages. Okay? Macrophages are quite big. They are specialised phagocytes. So they're still doing that engulfing. They take slightly longer because they have a little bit of a different job. So what they're doing is they're not working to break down all of the pathogen and then just dissolve it into the cytoplasm. Instead, what they're doing is they're doing the hydrolysis. They're using those lysosomes and the lysozymes to break down the pathogen. But then what they're doing is they're taking the pathogen antigens, so the proteins that we've broken off of the outside of that pathogen as we've digested that cell, and they're going to incorporate them or bind them to the antigens of the macrophage. Okay, so if this is my macrophage, I'm going to put it here, macrophage, and normally the antigens are just little circles. If I've got a pathogen inside that I'll draw in red, like this, and it's got little triangles on the outside, the way that the macrophage works is by digesting that pathogen and it's going to take those antigens from the outside of the pathogen and put it on the outside of itself. Okay? And what that does is by putting the pathogens on the outside of the cell, it produces something called a major histocompatibility complex, an MHC. Okay? Now this display of the antigens on the plasma membrane, on the cell surface membrane, means that that macrophage is now no longer called a macrophage, it's now called an antigen presenting cell. It's presenting the antigens. You know, it's fairly commonsensical, hopefully it shouldn't be too challenging for you to remember them. But those antigen presenting cells are then used in specific defences. Okay, so when we go into the next lesson, we start talking about B cells and T cells. That's what we're talking about. We're taking those antigen presenting cells and we're using them to trigger a really good specific immune response. Another way that macrophages and phagocytes work is by producing something called opsonins or opsonins. And these opsonins are little molecules, as you can see in the diagram, the little red bits, that tag or bind to the pathogen and they make them more visible. A bit like a lighthouse. They add like a little beacon, chemoattractants say, that help draw the phagocytes to the pathogens and increase the rate of phagocytosis. You don't need to know too much about it, you just need to know that it occurs. Hi guys, so as you can see, this slide does not have any exam questions on it because I haven't done it yet. If you're in my class, could you please go to our class notebook and then look at the content library for lesson 2.16, and somewhere on that file there will be either a link or a file of exam questions for you to just practice going over the non-specific defences. Mr. Mahita's group, hopefully he will have done it too, and if not, just ping me an email. It's Lauren Cherry at University of Kent Academies Trust, and I will send you the file myself. Okay, I hope you found this useful. Let us know if you've got any questions, queries, don't understand something, or just generally want a chat. Yeah, I'll be here. Yeah, see you next time. Bye.